All righty. I think we're good to get started. We still have a few participants just streaming in here, but we can get going. So good morning, everybody. Thank you so much for joining us today. My name is Kendra Wagner. I'm a research scientist with Rainbow Ecoscience. And today we're going to be talking about bacterial diseases in the landscape and arboriculture and best practices for tricky problems. As I said, I'm a research scientist with Rainbow. I know a few of you already. And I'd like to introduce today Dr. Matt Borden. Dr. Matt Borden is the plant pathologist at Bartlett Research Laboratories in Charlotte, North Carolina. He has practical experience in orchard and botanical garden pest management, plant problem diagnostics, and writing for industry publications. He gained a deep appreciation for integrated pest management while at Virginia Tech, followed by studies in landscape health and sustainable management strategies at University of Florida. So before we get going and before I hand it off, I just want to remind everybody that the chat is disabled for this webinar. So if you have any questions, comments, or concerns, please go ahead and pop it in the Q&A box at the bottom of the screen. And with that, Matt, I will hand it over to you.
Outstanding. Good afternoon, everyone, or at least those of you in Eastern Standard Time with me. So share my screen here. There we go. Kendra, look good?
Perfect.
Awesome. Thank you all for being here today. When Patrick Anderson asked if I would do this seminar a couple of months ago, I kind of laughed because our industry is sort of notorious for not having very many great options for bacterial disease management in the landscape. So I said, sure, let's do it. Let's see what we can find and have a good discussion about what we do have that's available. So this is my outline for today. I'm going to spend very little time on the individual diseases. And the reason for that, excuse me while I move something around here. There we go. The reason for that is that, these bacterial diseases are quite complicated, and there are a lot of outstanding resources out there from, YouTube seminars, other industry talks, extension fact sheets on the individual diseases. What we don't have a lot of is resources specifically for our industry in terms of management and the differences between recommendations for agriculture versus landscape and arboriculture. So I plan to spend as much time as possible talking about the tools for our industry, and then also a lot of options out there that might come into our industry in the future, but maybe are not quite there yet. Because what I've learned is that if we are not asking for these tools, the companies are not going to provide them for us. So without further ado, let's jump into the basics, and that would be your basic bacterium. So I love this chart. It just sort of shows a general size comparison. There's the little bacterium, and you've got the head of a, let's see if my pointer works here. I think it should, laser pointer. There we go. You've got the head of a nematode piercing into a plant cell, fungal mycelium, and then little bacterium. Even smaller than that are the mollicutes, which we are not going to talk about today, but that is a general term for the group that includes the yellows diseases and some others that are sort of bacteria adjacent, but that's a whole nother topic. So we're going to save that for another day. What's really cool about bacteria is that A, they're ubiquitous, but then B, just their reproduction potential is absolutely incredible. Under optimal conditions, this is a generality, but under optimal conditions, bacteria, given the resources and right temperature and moisture, can reproduce about every 20 minutes. So, over the course of a 24-hour period, you can have a number far higher than I know how to pronounce. But that is what makes them so prolific in the landscape. So this is how they get into plants. They are not the same as fungi, which a lot of the fungal diseases we manage have very active mechanisms for infecting plants, like having the aprosorium, which sort of pierces its way into the leaf tissue. Bacteria don't generally have those kinds of tools. Rather, they get into leaves through stomata or lenticels, which are natural opening in the plant tissues, or wounds of any kind, which is a big deal, Hydothodes, those open pores at the edge of the leaf, pictured here. That's a nice example of guttation out of hydothodes. And then nectaries and other openings. So they're a little bit more opportunistic, and if there is an opening, they have an opportunity to get inside the leaf. So in our industry, there are certainly a couple of major bacterial diseases, excuse me. We refer to them typically by the symptoms they produce. So leaf spots, vascular wilts, scorch, galls, yellows, blights, et cetera, et cetera. Compared to agriculture, we honestly have it really lucky. You know, some of these diseases are certainly problematic, and you might lose plant material, but compared with losing hundreds of millions of dollars in, region-wide crop loss, like is often experienced in the agricultural industry, we really have it lucky. So there is that. I don't want to downplay our problems, but the reason why our industry gets very little attention for bacterial disease management compared with agriculture is that it's just entirely different stakes at play. So one that is crossover affects agriculture absolutely, but also us in managing landscapes is fireblight. This is a major issue for malus, apple, crabapple, also a number of pears, and other rosaceous hosts. This does not affect the stone fruits, which I've found over the years is actually a pretty common mistake. People think they have fire blight in their peach every year or things like that. That is a different problem, often Pseudomonas, but it's not fire blight. So in orchards, there have been many, many years and many, many tens of millions of dollars that have gone into fire blight management in orchard settings. So because of that, fire blight management in orchards is highly structured, regimented, fine-tuned, because it's a huge deal. And we have some of those tools at our disposal, but not most of them. So really, it comes down to often trying to find resistant cultivars for your region, which is easier said than done, but I'll talk about that in a minute. The life cycle of this disease, if you don't know it, you have these perennial cankers on the infected host that in the spring produce a sweet, gummy mass of bacterial ooze, which is then picked up by insects, particularly flies and bees, which then land on the flower. And that is the infection court, where those bacteria get on the stigma of the flower, move down into the flower head, and then from there down into the spur, potentially into the twig, and then you end up with a canker issue. So it's a very difficult disease to manage, both for us and for agriculture. Another one that I see all the time in the landscape is crown gall, Agrobacterium radiobacter. Sometimes you still see it as Agrobacterium tumefaciens. I usually see this on euonymus, but cherries are another big one. In fact, I have a bag on my desk full of crown gall that I took off the root of a cherry tree. Typically, we remove these if they're branch cankers or planting species that are not susceptible to them. Here's a really cool example from ash tree, I believe, and you can see the crown gall there all around the major roots. Not a whole lot we can do about that. I'll come back to crown gall management later on. Third major issue in the landscape is Pseudomonas. This has a very wide host range, and that's primarily because the Pseudomonas taxonomy is very, very complicated. I don't understand it at all, but I do understand this concept of pathivars. So instead of, genus and species like we have in plants and sometimes subspecies, it gets very complicated when you're dealing with these bacteria. So we have phylogroups and many different pathivars that affect different hosts. So these generally cause twig cankers, shoot blights, wilts, leaf spots, and there's a lot of plants that are susceptible to these various pathivars. I love this photo of the twig at the top. You can see those are lenticels, so natural openings in the plant tissue, not wounds, lenticels. And the bacteria has infected at every one of those lenticels. Very cool. And then there's one at the bottom as well that's clearly on its way to girdling that rose stem with a canker. Fourth major one to mention is BLS, bacterial leaf scorch. This is probably very familiar to many of you in the Mid-Atlantic region. And in our industry, we primarily are concerned about oak, sycamore, a few maple, sometimes ash elm, but it affects citrus down in Florida, a different group of the bacteria. And then there's a lot of other minor hosts, which we do see occasionally and diagnose here in our diagnostic lab. But it's kind of rare to see them out in the landscape. But yeah, the ginkgo can be infected, for example, honey locust. It's a huge deal in olive over in Europe. And there's a variety of others. So this is when you see like an oak tree that starts to become very scorched around midsummer, July, and then very rapidly the canopy browns out later in the season and the leaves drop. That's typical symptoms of BLS. This is a chart I pulled from another slide set I had showing the different subspecies of Xylella. And this is kind of important because this is the only one that we are typically concerned with in our industry. So Xylella fastidiosa multiplex. This is the one that gets in most of our woody plants that we are concerned about and is also in a lot of weeds. Whereas if you got oleander bacterial leaf scorch, that's a different group. Down in Florida, it's a different one that is in citrus. And then it is fastidiosa that is a huge issue in grapes, and that is called Pierce's disease in grapes. Major, major issue in grapes. Okay, so that's it as far as specific diseases. How to diagnose these? I mean, if you have something like fireblight and you're very familiar with what it looks like, you can make a pretty accurate guess at that diagnosis. But beyond that, bacterial diseases are really quite complicated to diagnose. So this is a planting in Florida. Back when I was a young intern at the diagnostic lab with Carrie Harmon at the University of Florida PDC. So we were told, diagnose this issue in this planting as a project of sorts. So, you've got some infection in this plant. There's a yellow halo. It's clearly moving in from the leaf margins. So where do you start? Typically with bacterial diagnosis, you start by taking a piece of that tissue, putting it on a slide, and observing what is called streaming, which is exactly what it sounds like. You're looking at bacteria under vast populations and pressure sort of streaming out of that tissue. Depending on the plant, this might be done with a section of leaf tissue, as you see here, streaming out of the leaf tissue. Or you can cut a stem and see bacteria streaming out of the stem, which is something done for tomatoes, for example. Pretty cool. But beyond that, it gets very complicated, and this is why bacterial diseases typically need a very good laboratory to diagnose them properly. There's a whole range of tools used to narrow down what bacteria you're dealing with. These are just some examples of the common types of tests. So aerobic activity, does it grow in oxygen or deprived of oxygen? HR response, looking at how the plant responds to the bacteria. That's a pretty cool test. It uses tobacco leaves typically. Or King's B medium, so that's a medium that helps detect pathogenic Pseudomonas, which fluoresce under black light. So that's very cool, very fun. Other tests, again, it's all about whittling down, is it this group or that group, this group or that group? And then really it depends on what you need. So if all you need is a general diagnosis for management purposes, that's one thing. If a scientist is trying to drill down into something more complicated like subspecies or pathovar, it rapidly gets much, much, much more time consuming and difficult. But there are a lot of ways out there that the labs will use to figure out what bacteria you have, and it's quite different from identifying fungi, for example. Okay, let's get to the fun stuff, management. So I pulled out every book that I had around here about bacteriology, and I was curious what portion of the book is devoted to management, because in our industry, that is typically what we care about the most, right? Like learning about the biology of the organism is great, and the life cycle, that's great. But ultimately, most people want to know, what do we do about it? So this was a fun example. This is a Fundamentals of Bacterial Plant Pathology. It's a very good book. but the control section is only about 16 pages out of 342. And that's a very common trend as I went through a lot of my books. The control chapters are often some of the shortest chapters in the book, which doesn't bode terribly well for us because most of those control sections are written for, again, agriculture. Nevertheless, we glean what we can to apply to our industry. So, gleaning what we can, there are a swath of cultural management strategies that are quite appropriate for managing bacterial diseases, some of which we can use, some of which not so much. So let's just churn through these real quick. Resistant cultivars, I'll get to that in a second. That is something we can use for some diseases, but often the information just does not exist. So I'd call that optimistic. Managing fertilizer, that is something we can certainly do. Starting with clean plant material, this is a big one. Nurseries often have quite regimented programs for producing clean plants, and that is very important. Be very careful to buy plants from reliable nurseries that have good sanitation and disease management practices, because it's easy to buy a plant that has these essentially incurable diseases and accidentally put it out onto the landscape. Managing irrigation is a huge one, same as fungal diseases in the landscape, but often we don't have control over that, or you might have partial control or be able to advise someone, but sometimes we have no control. Letting the soil dry out is important. Mulching to manage splash and inoculum, same as fungal diseases, you all know this. Removing the whole plant is sometimes necessary, of course. Cleaning up debris, sometimes we can, sometimes they want to leave it. Crop rotation is a huge one, but we don't really do the equivalent of landscape rotation, do we? I mean, if you have a vegetable garden at home, crop rotation is easy, great. But if you have a landscape, not so much. I think the nearest thing we have is changing out of annual beds. And yes, you could rotate that if you are a mine, but typically those rotations are solely for aesthetics and not so much for something like bacterial disease avoidance. And then another one is wind management. So there are two things here. One is reducing the spread of wind-driven rain through the landscape. So having barriers, so maybe a tall shrub in between low beds of susceptible plants, for example, to break up that wind-driven rain movement. That is said to be able to help quite a lot. And then wind management to avoid wounding, so avoid twigs and leaves bashing against each other. That sort of thing creates microabrasions, which can be prime wounds for bacterial entry. So resistance is an excellent choice, like nothing better, if it is available. And unfortunately, in our industry, this is very rarely the selection breeding priority for ornamentals, right? We're selecting for shape, form, or leaf color, or flower color, bloom period, all kinds of things that we select for before disease resistance. So you will sometimes find these, but it's not terribly common and not nearly as common as I would like. So, for example, you can, if you want a crabapple variety that's resistant to fireblight, there are a variety of charts out there where you can select something. But you have to keep in mind what diseases are most prevalent in your area. So Radiant, I just grabbed as an example. It's a nice crabapple cultivar. It is very resistant to fireblight and powdery mildew, which is great. But it's very susceptible to apple scab and cedar apple rust, which is not so great. And so depending on which diseases are really severe in your area, that may be worthwhile, or it may result in extra fungicide applications to manage those other issues. Another example, so Pseudomonas syringae, that bacterial blight canker, most lilacs, common lilacs, are very susceptible. Anecdotally, you can find a little bit of information to say that some cultivars are less severe than others. And then there's a handful of trials to show that some of the other alternative lilac species are much less severely affected by this bacteria. And if you really dig for this kind of information, you can often find some, but again, it's just not very practical for most diseases we deal with. Either the information's not there, or no one has ever put the plants through a screening process for the exact bacteria you want to know about. Nevertheless, it never hurts to look for it and taking a few extra minutes. Um, but cultivars, it depends on what the nurseries are growing. So even if I wanted a pink Elizabeth lilac, for example, here, I would have to be able to find that in the trade from a nursery that is able to sell it. So yeah, these are issues that we're all used to, right? Next, managing fertilizer, big one. Um, Pretty much any bacterial disease management section you read about will mention this. Avoiding excess fertilizer is primarily in reference to nitrogen. Now, we tend to generalize and say nitrogen. It is worth noting that there are different kinds of nitrogen in fertilizers, right? There's ammonium nitrate, nitrites, etc. Those can have distinctly different effects on diseases, but in general, we want to avoid excess nitrogen, especially from about midsummer onward. And the reason for that is that if you are accidentally inducing the plant to grow more late in the season, those tender tissues tend to be the most susceptible to winter injury, frost damage, and those injuries become wounds in very susceptible tissues that are prime for bacterial infection. So that's the rationale there. I also threw this chart up here. There's a lot of information out there from agriculture about how different nutrients and micronutrients relate to disease management. And we are very young in our industry in terms of how those relate to the different ornamentals we work with. But it is worth noting that in general, as a complete top level generality here, things like potassium have been shown in studies, again, in agriculture, but we can infer a little bit, to generally have a decreasive effect on bacterial disease if there is sufficient amounts of potassium. So right here, bacterial, 5 studies saw an increase in disease and 21 studies saw a decrease in disease. Now, again, this is like a meta-analysis type overview. You would have to go in and pull the information out of all those studies to figure out exactly how it related to your situation. But these kinds of things are very helpful for looking for general trends, right? So don't go overboard with the nitrogen. Make sure there's enough potassium. Be a good summary here. And then back to the issue of wounding. There are a lot of products out there advertised to paint or spray on wounds. Personally, I don't use these, and I don't know many people that do. But if this is a concern, they are available. Really, the best thing to do is to prune when conditions are not good for fungal and bacterial disease infection in open wounds. So winter pruning when it's dry, right, is optimal. But nevertheless, there are products out there that are specifically designed to help provide some sort of barrier if you are pruning during wet spring weather when it's almost certainly going to get infected with something, right? So can't really say how well these work, but they are available. This is another one that that is available, and I think we could use a little bit more in our industry. So a lot of work has gone into building models that are available to the public, primarily growers, for certain diseases. Boxwood blight is one. I use that one regularly. Another one is fireblight. So there is this uspest.org website where these models are located. You can subscribe based on your location, put in your zip code, find the accurate weather station nearby, and then they will send you, or you can get it off the website, tables to predict if you have a major infection period coming up. So for example, I pulled up the chart for March of 2024 and 2023. So the dark bluish purple line here was the weather in 2024 and the light blue was 2023. So this is the infection period for fireblight specifically based on the kinds of environmental conditions it prefers and also bloom period, right? So we saw, some low to moderate risk of infection in middle of March 2024. Compare that with April, however, and you see there's extremely high risk of infection for much of April. And how would we use this? Well, we would use this as a forecasting tool to be able to see about a week in advance is probably all it is in terms of accuracy, to go out and put on the preventative applications we need on those tissues before we hit these exceptional and extreme risk infection periods. So they might be useful. Gail Durham just put a note in the comments saying Diseases in Trees and Shrubs, second edition by Sinclair and Lyon is the go-to book. Couldn't agree more. In fact, I've got it right here. Yeah. If you don't have this, it's absolutely worth getting. Sorry to move away from the presentation for a second, but that is worth saying. It's a very affordable book, outstanding resource. Okay. So this is a neat product for crown gall. I have never used these products. I don't know anyone that's ever used them, but they've been around for a long time. So crown gall, produces a tumor-like growth. It is almost entirely originating in the nurseries, that when they're making cuttings and such, those wounds get infected with this bacteria that eventually causes gall-like tumor tissues. So gall troll A is one that is supposedly able to pre-infect those wounds with a non-pathogenic strain of the bacteria that colonizes it but doesn't cause disease. And then Gall X is another product produced by the same company that is essentially a mixture of hydrocarbons that selectively penetrate the tumor-like cells of these growths. Again, I haven't used these personally, and I don't know many that have, but they are labeled for crown gall. They're some of the only things labeled for crown gall. And Gall Troll A would be preventative for the propagation process. And then Gall X would be if you are trying to cut off a growth of crown gall and then paint on this mixture to prevent it from regrowing. If possible, though, if it's on a branch or something, you would just prune it out, right? Yes, we'll get the name of that book up in the chat, sure. So moving on to something a little bit more powerful, antibiotics. Streptomycin was discovered as an antibiotic produced by a bacteria, which happens to be the state microbe of New Jersey. Fun fact for you. So Streptomyces griseus produced what we now know as the antibiotic streptomycin. This was phased out of most uses because bacteria are exceptionally good at developing resistance to anything thrown at them, including many of these antibiotics. So while they're still used quite heavily in certain areas of agriculture, we don't typically use these much in landscapes. Nevertheless, I will mention one that is available. So agromycin 50 is a streptomycin sulfate available for ornamental plants and shrubs. To my knowledge, this is the only such streptomycin product labeled for our industry currently. Don't quote me on that, but it's the only one I know of. It's not a silver bullet, right? These antibiotics are exceptionally good tools when used properly, but resistance is a real risk. And then also, it doesn't last very long. 3 to 4 days is what the company says. So to effectively have good coverage from this antibiotic during the entire bloom period of, say, a crabapple, to protect it from fireblight, you might have to be applying this, well, as it says right here on the label, continue applications every 3 to 4 days during blossom time, and then apply additional sprays every 5 to 7 days after bloom period when weather favors the spread of fireblight. So this is something that can be done in orchards where they are out there doing cover sprays very heavily during this time of the year. It is not typically something that we can, or honestly should be doing in most landscape situations. I don't know what all of your situations are, but I know that most people in our industry are not able to visit a client every 3 to 4 days from March through April to do an application. It's just not logistically feasible. Another antibiotic in our industry is OTC, oxytetracycline. This was also derived from a Streptomyces bacteria. This is very effective as a suppressive agent for bacterial leaf scorch. Does not cure it, and that is an important distinction. It only lasts about 1 season to offer suppression, so knocks that population of Xylella down in the host plant. There's a lot of suspicion out there amongst our pathologists that there probably is widespread BLS resistance to OTC, but no one has really looked at that yet. So if you see a lack of response to BLS from your OTC injections that you've made year after year after year, it could very well be resistance. Just we haven't really identified that yet. Sorry, I'm seeing some questions pop up in the Q&A, but we'll get to those at the end, don't worry. All right, I'm going to spend a few minutes on this, our number one chemical tool, copper. Copper is often misunderstood, so I wanted to highlight a few of the pros and cons and how to use it effectively. So one of the pros is that it's a micronutrient, so plants do need copper in very, very, very small amounts, but it's rarely ever truly deficient in the situations we're working with, so we don't typically need it as a micronutrient. And applying it too much can risk excessive buildup, of course, and non-target effects. Another pro, a lot of people prefer natural or OMRI organic options, but remember, organic does not mean safe, nor does natural does not mean safe, and copper is no exception. So while it's a powerful tool, it is also very highly toxic to aquatic life, beneficial microorganisms, can be quite persistent, of course, as a metal. So we have to use it wisely. Another pro is that we use it for dormant season applications. In particular, we often use a high rate of copper mixed with a little bit of horticultural oil as a dormant season cover spray to knock down a lot of diseases on twigs and bud break and that sort of thing. Very good tool. And one more con is that it can be reactive, and sometimes people get in trouble for spraying copper and it hitting other masonry or metal around homes and causing corrosion and problems. So I got a couple of slides from information that I largely pulled from a really good slide set by Carrie Peter. She's a fruit pathologist at Penn State and has done a lot of work on copper. So yeah, credit to her for the next couple of slides. But copper is a general biocide. That is why it's used as a bactericide and a fungicide and an algicide, etc. It is not really curative. It acts as a protectant, so you have to apply it pre-infection. And it sticks where it hits, which is a good thing generally. It does not redistribute itself either inside the plant or redistribute itself on the surface of the plant. So while on the leaf, it requires moisture to be present in order for it to be active. So if it's a dry day and there's a copper residue on the leaf, it's not really doing anything. But if it has a light rain the next morning, well, that's probably when the bacteria are also active. So you want the copper to be active, and that's how it's working here. The copper products that you spray on the plant are gradually releasing copper ions. It's those copper ions that are working for us to knock out certain fungi and bacteria and such. So again, it's most effective when it's working against diseases that need that free water present to develop. And it essentially kills a lot of these things by messing up critical enzyme functions. So the challenge with copper is that you want to have enough present on the plant tissue to kill the target, in this case bacteria, while keeping the concentration low enough that it is not going to cause injury to the plant. So it's a constant balance. We want enough copper to work, not too much that it causes phytotoxicity. this has to do with solubility and pH, and it's important to understand this. So if you have a copper that is extremely soluble, that means that all those little copper ions are going to be available all at once when the leaf surface is wet. And as such, they're going to be washed off quite easily if it rains a lot. On the other hand, if you have an insoluble copper or a fixed copper, those are on the leaf surface that dries, and it's much, much more persistent. They don't readily all redissolve when the leaf surface gets wet, and it's a slower release of copper ions. So you have longer residual activity and less phytotoxicity. So there's a lot of copper products out there, particularly for ag, but also for our industry. So two that we use in my company and a lot of others are copper hydroxide and copper oxychloride, which you'll see right here. So this is the water solubility chart. Over here, the fixed coppers, and then copper oxychloride is somewhere in the middle, sort of an intermediate, and then copper hydroxide is over here, more soluble. So for example, if we look at this product badge, which is one that we use, the copper oxychloride is going to have that trait of being a little bit more persistent and lasting longer on the leaf surface, whereas the copper hydroxide is more soluble and will have a more rapid activity and release of those copper ions. So that is why you often see these copper products with different formulations of copper in it. It's because they have slightly different traits, and one is often longer lasting, one is often more readily available. And this has to do with the pH of the spray solution. So if you read these labels, it'll often say, be very careful not to apply in a solution of a pH less than 6.5 or something like that. And that is because if you have it in an acidic solution, it makes those coppers much, much more readily soluble, which reduces your persistence and increases your risk of phytotoxicity to the plant. So we want to avoid that. Last interesting point with coppers is that we're used to looking at the active ingredient when we read labels, but when it comes to something metal like this, it is the percent metallic copper in the solution that is very important. Vegetable growers, for example, will often look for products that are 20% metallic copper or more. And you see that they're not always the same. So, Mastercop, for example, 21.46% active ingredient, but only 5.4% metallic copper. Whereas that badge product I mentioned a minute ago is 20% by weight. So it's worth keeping in mind that if you're reading these and you're wondering why it's talking about the percent metallic copper, the higher the percent metallic copper, the more of those copper ions will be available. That's a good thing. And then lastly, phytotoxicity risk, don't mix with anything acidic. As I said, this includes the polyphosphate products or phosphonate products like Reliant, Agrifos, polyphosphate 30. Also, don't mix these with thiophanate methyl. I don't know how many of us are using Clear E3336 anymore, but that or Topsin M. And then most people say don't mix with anything mancozeb based, although in agriculture, there are some situations where they do mix copper and mancozeb. So it's a calculated risk for them, but we don't recommend that. Avoiding contact with metal, I mentioned that. And then also plants differ widely in how sensitive they are to copper. Some cultivars of peach and that group are much more sensitive than others. So be careful. Okay. In the last few minutes here, I want to look ahead into the future and talk about some tools that we may have access to in the future. And first, remember that bacteria are everywhere. Only a very, very, very tiny percentage are bad. There's plenty of good bacteria out there that we can harness as valuable tools. There's the whole swath of bacterial insecticides, the antibiotics that we've derived from bacteria, as I mentioned. This is a pretty prominent one. Bacillus subtilis, strain QST713. These are in the Serenade product range. In our industry, it's cease, which That's on the next slide. These are available for both professional use and homeowner products. As far as biological control agents, I think this type of Bacillus subtilis product is probably the best known. And these generally have good results. However, good results mean different things to different people. If someone is expecting complete and utter control, like a 100% efficacy of a product, then as with most other biologicals, this may not be sufficient. Nevertheless, it's worth looking at. These generally have good results. Cease is one product that is labeled for our industry, and there's others out there labeled for homeowners, etc. So this is a live bacteria. It colonizes the plant surface and works to outcompete some of the other pathogenic bacteria. Direct antagonism. And you'll see there's the cease label. It is labeled for Erwinia, which is fireblight causing bacteria, Pseudomonas, and others. But always read the application interval. As with many of these biologicals, is applications on a 3 to 7 day schedule possible logistically? For some people, yes. Many others, no. Blight ban is an interesting one. I am not aware of a label containing Pseudomonas fluorescens A506 for our industry. As far as I know, this is really just ag use. But this is a strain of bacteria that is a non-frost forming strain. So a lot of the other pathogenic Pseudomonas cause damage by inducing frost damage. So frost is more likely to form, and ice nucleation is more likely to form when that pathogenic bacteria is present. This one is a non-frost forming strain of the same general group of bacteria. So sometimes growers will spray it prior to freeze events. It will colonize those tissues and prevent the growth of those ice nucleating Pseudomonas. So it's a really interesting product. It's not labeled in all states, but worth mentioning. This is a really cool one. You might hear about this on news or something. I've seen a number of articles because it's a fun topic, but some years ago, a company called B-Vector Technology, and I'm sure there have been others, had this very bright idea of having the bees do some of this work for us. So we know that bees may accidentally spread Erwinia bacteria from flower to flower and cause fire blight, right? So why not make the bees carry the antibiotic on their feet and carry that antibiotic from flower to flower to flower instead of the fire blight bacteria? Really cool idea. So they have tried this with streptomycin, with Bacillus subtilis. I've seen it being used experimentally with trichoderma in strawberries in California. I think their current product is using only Clonostachys rosea, which is another beneficial microbe, but it's a great idea. And I don't know if we will ever see a landscape equivalent to this, but it is cool. So here's me working at an experimental research station in Virginia. We actually did work with these products. So we got these bee boxes. In this case, they were bumblebees, and it's a little hive that you get in the mail. And then the bees, in order to enter and exit the hive, have to walk through whatever powder solution you put there. In this case, I believe that was streptomycin powder, so an antibiotic. And then the idea is the bees go out into the orchard, carry that antibiotic precisely to where it needs to go, and saves you from having to spray the entire area with that product. So in theory, this is a really cool idea. Be nice if we ever saw something similar in our landscape, but I don't know. And then in this case, we picked all of those flowers and sprayed them with fireblight to see how well it worked. Here's another, and I'll wrap up very shortly here, but ASM is a very interesting product. Actigard by Syngenta. This is a powerful synthetic inducer of systemic acquired resistance, so it upregulates some channels of the plant defenses and helps plants defend from infection and penetration of bacteria. Unfortunately, it is only in agricultural use currently. And a lot of us have been asking, will we ever see one of these labeled for ornamentals? The answer so far, it seems like Syngenta is not really wanting to pursue it. But this is a fascinating product. There's a lot of research showing that this Actigard alone or in combination with other things can be very beneficial in defending against a lot of bacterial diseases. Interestingly, there was also a paper from University of Florida recently showing that it was quite helpful in reducing rosette virus disease. So again, very cool product. And then lastly, bacteriophages. This is cool. Genuinely, it's just fascinating. Bacteriophage are viruses that attack bacteria. They're probably the most abundant type of organism on the planet. They seem to outnumber bacteria about 10 to 1. But the idea is that we could harness some of these viruses to attack the pathogenic bacteria, infect them, destroy them. Truly a biocontrol wonder agent is what it sounds like. In reality, we've got a long way to go. So Agrophage is one product out there labeled for fireblight. I just checked with the company last week, and they seem to indicate that it could be used in fruit trees, in landscapes and residential, because it doesn't exclude that. Don't quote me on that, but that's what they said. Nevertheless, this is a bacteriophage product, a virus being used to attack Erwinia bacteria. Very, very cool. As with other products, though, that I've mentioned, the application interval is really the kicker. It may not be logistically feasible for us. Also, it has to be refrigerated. You can't put it in your pesticide shed. So apply as a preventative to protect the growing tissue, as a preventative when conditions are conducive to heavy disease pressure, all bloom stages, and as a curative. Yeah, that's going to be a lot of applications, unfortunately. The real problem with these foliar applied phage is that they are very, very rapidly degraded by sunlight. As soon as UVB starts to hit them, they just disintegrate. Yeah, this is a huge obstacle. There's not really any way around it. So they are exploring ways to apply these phage with other solutions to protect them from UV degradation, like kaolin clay and other products. But we've got a long way to go. Yeah. Still, it's very cool. There's another company out there that has produced a different cocktail of phage for a xylella, which you may remember also causes BLS in our trees. This particular phage attacks the strain of xylella that causes Pierce's disease in grape. And I'm not allowed to say anything more about this project, except that this should pique some interest for having a similar product to look at BLS in trees, for example. We'll see how that goes. And very lastly, what about potassium phosphate? This is often a recommendation for assisting with bacterial disease management. To be honest, it makes sense that these would help, but there's very, very little data directly showing that bacterial diseases are significantly reduced by potassium phosphates. They may be, but again, there's just, we need more research in this area. So if you have anecdotal stories, please share them with me. All right. So in summary, there's an adage in bacteriology that you should take home and remember. If it dries, it dies. If it dries, it dies. These bacteria are extremely plentiful, but they're also rather environmentally sensitive. If your day is hot and dry and sunny, your pathogenic bacteria are going to die, which is a good thing. Summary point number 2, use copper carefully. Summary point number 3, if you can, avoid antibiotics. They're powerful, but should not be our go-to. And then remember those cultural management tools. Use clean tools, buy clean plants, work when it's not super susceptible to bacteria exploding on your wounds of plant tissue. And diversify your landscape if possible, of course, is always a good general recommendation. Avoid monocultures. And then if you think you need to diagnose a bacterial issue, you've got to send it to good qualified diagnostic laboratory. So that is my summary. And I'm going to stop sharing so that we can get over to some of these Q&A questions. Thank you.
Thanks, Matt. That was awesome. I really appreciate that talk. For those of you who had questions, there will be a recording available for this webinar on our website tomorrow. So if you missed anything or if you were late, you can hop on our website tomorrow and check it out. So we do have some questions. Just a reminder, the chat is disabled. So if you do have any questions, please pop them in the Q&A and we'll go through them live. So the first one is more of a comment from Tyler. He says, I honestly did not know there were state microbes. Interesting. Thank you for that, Tyler. The next one is from Michael. Is there any solution for slowing the spread of crown gall in black cherries or black oaks?
Not that I'm aware of. I don't know anyone that has successfully tried those products on existing trees in the landscape. I'm sure some people have attempted to cut out the galls, but I really don't know how well that influences plant health. So not that I'm aware of.
Okay, thank you. Eric asks, why can't streptomycin be injected like fungicides?
There have been injectable streptomycin formulations, yes. I'm not exactly sure why there are not any currently, except that the oxytetracycline is from what I've read, much more active against Xylella and that group of bacteria. So I don't think it's a case that it cannot be formulated as an injection, but that there are no products labeled for that on the market. Certainly not for us. There are a lot more injectable antibiotics being evaluated in citrus for those much more economically important agricultural diseases.
Thank you. Brody asks, should you inject OTC yearly or every other year? I'm concerned about injection site wounding.
Kendra, you might be more qualified to me than answer that one.
So with that, it really depends on what species you're looking at. Something like palms, you have to be careful with, and it depends on what you're trying to treat. That would be really case dependent, in my opinion. We do have some clients that will do OTC injections several times a year, and then some of them, again, case dependent, depending on the species and what you're treating for, they'll treat every other year or so on and so forth. So it really is case dependent. And if you have any more questions on that as well, I think that was Brody who asked that question, don't hesitate to reach out and I can help walk you through that as well. So Craig asks, does mixing copper products reduce pH of a solution compared to original water pH?
That's a good question. I would hope and assume that all commercialized copper formulations are formulated so that when they are mixed in with neutral or near neutral water, they produce a solution in precisely the right pH range. Maybe that is an unsafe assumption, but I would hope that that is how they're formulated. So as far as I know, if you start with neutral or near neutral water and then mix in your copper, it will be the correct pH. But I certainly have not gone around checking all of those solutions.
Yeah. Okay, thank you. Benjamin says, I have a palm with black streaking starting out of the interior of the frond and then spreads. I'm not sure if it's viral, bacterial, or fungal. Any tips on narrowing it down?
Send it to a diagnostic lab or send me photographs. I would be happy to at least look at photographs and refer you to a palm specialist based on what I see. No problem there. My email is mborden@bartlett.com. But yeah, we do a lot of palm samples and they can be rather tricky to narrow down even from photos.
Absolutely. Yeah, palms can be tricky. And we also have another palm webinar coming up in the coming months. If you go to our website and click on education, you can register there. And we'll be going through common insects and diseases seen with palms on the East Coast. So Brody says, also, what is your fireblight protocol?
Yeah, fireblight is just really, really tricky when you have very limited opportunities to be on a landscape and making applications. So our protocol For some trees, such as certain ornamental pears, we might do an OTC injection. More commonly, for something like a apple or crabapple, we're going to rely on a high rate of copper plus horticultural oil as a late bloom period bud break application, followed by a very, very low rate of copper that is appropriate for the bloom period. And that can work pretty well, but it's, none of these options are quite as perfect as you can get with a very, very good orchardist.
Okay. Thank you. Would a copper spray be less phytotoxic when sprayed when trees are dormant?
Oh, absolutely. Yes. That is why we can use a 10 times higher rate during the dormant season, as opposed to a much, much lower rate when a tree is in bloom, or sort of an intermediate rate when the tree has foliage on it. And if you read the label of your copper product, it should very clearly lay out those rates and state which one is appropriate for which stage.
Okay, thank you. Our next question is, did your experiment with streptomycin and bumblebees help protect the flowers from fire blight?
Um, we had to scrap that project, so I can't share the results.
It always works out like that, doesn't it? You go to study it and it disappears.
Yeah, sorry.
Okay, no worries. Okay, Victor asks, is there something to get better control on crown rot on cherries?
Crown rot or crown gall? I'm not sure about crown rot. I'd have to follow up with you on that to figure out exactly which disease is being referred to. Sorry.
Okay, we'll reach out to you, Victor, and follow up on that question. The next question is, does potassium phosphate truly stimulate SAR?
Potassium phosphate products absolutely do stimulate certain plant defense pathways. I can't remember off the top of my head if it's the same pathways stimulated by product like Actigard or not. Kendra, do you recall which pathways? I mean, it is very well researched.
No, I can't remember off the top of my head.
We
We can
have some
follow.
tech reports on the topic, and there are good resources out there. Yeah, it's not snake oil. There's plenty of good solid research showing that potassium phosphates can be very good defense-inducing agents.
We can follow up with you, Mateo, on that. Greg asks, how would you recommend sanitizing tools?
Yes, depends who you ask. Some people prefer heat, which is time-consuming, kind of dangerous, takes a lot of fuel. Most people would use a dilute solution of bleach, so like a 10% bleach solution, and by that they mean taking household bleach, which is already at like 3%, and then mixing that 1 to 9 parts with water, and that's their 10% bleach solution, which it's not really, but you get what I'm saying. But there are other professional products out there for our industry used in greenhouse pest management and such that are very good and slightly safer disinfectants as well. But if it's just a couple of tools at home, then a very dilute bleach solution is the recommendation.
Thank you. Julie asks, suggestions for bacterial cankers on trunks of trees.
Oh, that's a tough one. I have not really run across anything that gives us good options for managing severe bacterial cankers on trunks. No.
Thank you. Our next question is, what 3 trees would you pick for the best all-around disease resistance?
Hmm.
It's a tough one.
Ginkgo certainly comes to mind. Absolutely. I think it would largely depend where you are located for others. Coosa dogwood is pretty awesome if you're looking for a small flowering tree. Kendra, do you have any other favorites?
No, I was going to say ginkgo as well, and that's definitely a case dependent. It depends on where you are in the country. This is from an anonymous attendee, but whomever asked this, if you want to reach out, we can maybe talk you through some of these options. But ginkgo would be at the top of my list as well. Okay, Steve asks, any recommendations for bleeding bleeding canker on weeping cherry. Oh, sorry, Matt, just before you answer that, we are coming up on time, so if people have to hop off, please go ahead and do so. I did put up the ISA CEU code. This is for your records only. So when you registered, you should have put in your ISA certification number. If you didn't, please pop it in the Q&A, not the chat, just in the Q&A, and we'll make sure that you get credit for this course. But this code that's up here, there's no need for you to do anything with that. It's just for your record. So if you have to hop off, we appreciate everybody for joining today. And again, there will be a recording of this tomorrow on our website. And please take a look at our future webinar sessions and our in-person seminars as well. Sorry, just I saw people hopping off. I wanted to make sure we said that. So any recommendations for bleeding canker on weeping cherry?
Nothing comes to mind. Most people are going to prune it out and try to make a cut 8 to 10 inches below the canker. But other than that, I haven't seen any good solutions for well-entrenched cankers. It's not like Phytophthora canker where you can spray Reliant on it and kind of isolate it. The bacterial cankers are more difficult to manage.
Okay, thank you. Daniel asks, how important to sterilize tools between cuts in winter or dry summer conditions and injectables like Bacterial?
This is a controversial topic, believe it or not. I've known some PhD students that have specifically done work on this to measure how important is sterilizing between cuts, especially when, as you say, conditions are not optimal for disease transmission. In general, those projects find that transmission from pruning tools is far, far, far less likely than we really expect it to be. So when we say sterilize tools between cuts, that's more of a best management practice out of an abundance of caution rather than out of a body of literature that says you're going to get severe infection if you don't do this. So specifically looking at cutting out fireblight and such, as long as you're making that cut far enough below the canker, sterilization is probably not as critical as we do it out of an abundance of caution.
Absolutely. Okay. James says, any info/thoughts on VSD/CT?
Sure. So vascular streak dieback slash Ceratobasidium theobrome is an important issue in Tennessee nurseries and some other states in the Mid-Atlantic and Southeast now. I have lots of thought about it. It's a little bit off topic, but we have been looking at that issue a little bit, and I know Rainbow has a experimental product that has seen very good results in a study that is now published. So we're very hopeful that we may have a pretty good tool for it in the near future.
Hopefully. Gail says, excellent presentation, Matt. Thank you for the bacterial solutions that are out there, and they are hard to find. Thanks for that, Gail.
Thanks, Gail. Fireblight crabs.
Yeah. Okay. We have lots of questions here that just keep pouring in. We've got about 45 left, so I don't want to take up all your time today, Matt. If you have to hop off, we can follow up with some of these folks via email. Just let me know.
That's okay. Let's see how far we can get.
Okay, perfect. Okay. I'm going to hop actually down to what is the best way to find out the pH of water? We typically are using a municipal source. Would they have the information?
I haven't seen, or I have not been able to find a whole lot of information published by the municipal sources. I bet that does exist, but I struggle to find it. Honestly, I think there are really good pH strips out there available to purchase online. They're pretty accurate, and they'll get you within a half pH point or so, which, because it's a logarithmic scale, is usually sufficient. So honestly, if you find an extension bulletin that has tried a lot of pH meters, and you can find a whole packet of 100 strips for $10 or something like that, that'll probably get you a good solid ballpark estimate.
Okay, thank you. David says Bradford pears were once considered blight resistant, but now are commonly infected. Has this disease mutated?
That's an interesting question. A lot of the issue with Bradford pears is misidentification of the problem. Uh, I I have seen a lot of Bradford pears that are being treated for fire blight, and then you look at it more closely, and it is other issues that also cause twig blight on Bradford pears. So, first thing would be to double check that it is actually fire blight that you have, and not um, cedar quince twig canker or another bacterial disease like Pseudomonas, because those are often all sort of lumped in like fire blight because it looks very similar. Um, as far as mutating or such, I am not aware of any of that, but the people researching fire blight and ornamentals is also, well, there's not many of them out there. So
Yeah,
there's a lot of
yeah,
questions that we just don't know the answers to.
exactly. William says, is there a SE for plant growth regulators for fire blight and pear, for instance?
There has been a lot of research using a growth regulator called Apogee for fire blight management, which can be very effective. That is an agricultural product. I don't know that we have an exact similar one for landscape ornamentals. We do see some interesting results from Cambistat, paclobutrazol, for fire blight, and we're not exactly sure what's going on there, but there does seem to be an effect. So that is probably something that perhaps you all at Rainbow, Kendra, may be able to do a little bit of work on in the future, and we learn if some of the existing tools that we have may have some benefits.
Absolutely, absolutely. It's a good idea. Next question, does streptomycin control mycoplasma?
I don't believe so.
Okay,
Yeah.
thank you. Chantel says, I went to a customer's property this year to treat their ash trees via injection for EAB. I noticed a significant portion of the drill wounds from the prior injection service 2 years before had not healed over and were oozing. I did not do the prior treatment. Do you have any suggestions as to what happened, what might have been going on with them, and if anything should be done to address this issue? I've never seen this before in this situation. Thank you.
Oh, that's a great comment. Kendra, I'm sure you and your team have seen this as well. So
Mhm.
there are a lot of BMPs, best management practices, when it comes to drilling trees, and it really is crucial that the person doing those applications has had proper training in this. A lot of it comes down to sterilizing the drill bit, or more importantly, using a very, very sharp drill bit, not drilling too deep. That there's a whole series of BMPs that, if they are not followed, can increase the chances of infection at the site, or just decrease the likelihood that the tree will seal over that wound in a timely manner. So in pretty much every case I've seen similarly, it was really just a case of someone not having had the opportunity to go through proper technique and training on drilling trees. And they're just going in with a drill bit and doing the same thing that they would have done in their wood shop at home, which is a problem.
Absolutely. I like to say the equipment and the chemistry that you're using is really only half of the solution, but it's the application tools, methods, and training that goes into making sure that you're going to get success in
Okay.
that treatment. Dennis asks, how does Lysol work for sanitizing tools?
A lot of people use Lysol, and as far as I know, it works. Yes, I know landscapers and even some of our people that will keep Lysol on hand.
Okay. Just trying to go through some of these. Maybe this is a deeper question than the webinar is made for, but I'm an amateur in this field. What steps of diagnosis do I follow to get the answer of treatment?
Oh, that's a good question. So almost every state has a land-grant university system with a diagnostic lab, or some of them contract with their neighboring states that might have a lab. And typically, if you need a diagnosis, you contact your state's lab that handles that kind of sample. Make sure that they can do that kind of sample diagnosis for you, and then you would send it in and get a response back for, in most cases, a very affordable cost. Some companies like ours have our own large internal diagnostic lab where we do this kind of work, but in general, most people are going to want to work through their state lab, and it might be $10 to $20 to $25 typically, so not too bad.
Okay, thank you. Clarissa says, I'm curious about the topic of fertilizing during summer and late fall. I can understand avoiding fertilizing in the summer to prevent new growth that may be at risk from new tissue. However, I thought fertilizers in the fall can be a good prep and nutrient PP. So I think this was to prepare for winter.
Oh yeah, a lot of that is regionally dependent. I probably should have clarified that. If you are um, in, South Georgia or Florida, fertilizing in the fall means something quite different than those of you who have gardens in the Northeast where your garden is quite thoroughly dormant, much earlier in the season. So yes, yeah, you're quite correct, and applying a slow release fertilizer that isn't really going to be available to the plant till spring is okay. You're just trying to prevent any of that excess growth going into the cool season.
Okay, thank you. Who decides what biological control is recommended for the agriculture industry and other industries?
Well, largely the EPA, but a lot of it just comes down to cost. There are a number of companies working on these products because there's absolutely a need and a market for reduced risk biocontrol products, but it's very, very expensive, and then also just the nature of biocontrol products, it's rarely 100% effective. And so depending on the threshold and what the grower's tolerance levels are, it's just a challenging industry. Yeah. I would encourage you to go and reach out to some of these companies and find out how they're doing this kind of research. And yeah, it's interesting stuff for sure.
Okay, thank you. Um, what practices would you recommend for communities with low funding for trees to best mitigate disease, especially those along streets? Are there specific grants or cost sharing for this?
There definitely are grants. I don't know much about that process, but I certainly have seen proposals where municipalities work with Tree Fund or other organizations uh, to do surveys or understand their tree susceptibilities, or in some cases even get funding to do large scale treatments. So those opportunities are out there. Yeah.
Okay. Have you ever used hydrogen peroxide to treat a disease in trees?
Yes, there are a lot of hydrogen peroxide products out there, including some used in organic orchards. Um, they're very smelly products, and as hydrogen peroxide, anyone who's ever used it, it doesn't last very long. So it's sort of a rapid fire oxidizing agent. Um, as such, it doesn't really have specific fungicidal activity or specific bactericidal activity. It's more of bombarding those microorganisms with an oxidative reaction. So yes, I've used them, but it's not really something I strongly recommend. And they're also very broad spectrum. There's no control over which microorganisms you are killing and which you're not when you spray something like that.
Okay, well, the rest of the questions are just folks putting in their name and their ISA CEU numbers. So again, everybody, this code is just for your records. There's nothing that you need to do with it. When you registered, you should have entered your ISA CEU number. If not, please go ahead and pop it in the Q&A. And with that, Matt, we really just appreciate you joining us today, and we just appreciate your time. So thanks again, and it was good to see you.
It was a pleasure. Thank you all.
Thank you, everybody, for joining. Bye-bye.
Take care.